California sea lions, bacterial and fungal contributions to abscess formation. Dr. Krumbeck is the CEO and co-founder of Midog Animal Diagnostics in Irvine, California, and is leading the efforts to bring novel diagnostic tools to veterinarians to improve the diagnosis of infectious diseases in companion animals. Using molecular DNA sequencing to understand the animal microbiome, Dr. Krumbeck has published over 30 peer-reviewed manuscripts on the microbiome of pets in health and disease. She earned her PhD from the University of Nebraska-Lincoln in cell molecular biology with an emphasis on microbial gastrointestinal ecology. Dr. Krumbeck has studied the interaction between microbes and their hosts for over 10 years in order to understand how they specifically affect health and disease. She is passionate about applying the current knowledge of the pet microbiome to improve infectious disease diagnostics and promote good antibiotic stewardship.
Thank you so much. It's really a pleasure to be here. Thank you for letting me present some of our research data that we've been finding on the California sea lions who've been suffering from abscesses from both bacterial and fungal infections. And I actually don't know how to advance this slide.
The green button on
the green button, perfect. So as a disclosure, all of these are samples that were sent to Midog for animal diagnostic testing, and we did the study retrospectively on the samples that have been sent to us. So let's just jump right in. As you all know, sea lions are susceptible to abscesses, and these abscesses are the second most important cause for their mortality. And these are often worsened over time, especially treatment can be difficult with all the co-environmental factors. And I don't need to tell you that as clinicians, you know, the more that you know about the infection, the more successful your treatments are. The more you can identify what is causing the infection, whether bacterial or fungal infection or what kind of pathogen, the more targeted your treatments can be, ultimately resolving the issues faster. However, when these symptoms arise, we are relying on tools that are a little outdated. Culture testing was phenomenal at the time. It was first developed in the 1880s, Robert Koch and Louis Pasteur, groundbreaking idea that pathogens exist and hey, we're able to grow them in the lab in these little petri dishes. However, there are several limitations to this tool. I think a lot of you know the no growth results. They are very, very common. According to the Iaaam, a study that was published a few years ago, 30% of all culture tests being submitted by veterinarians return no growth, period. So that's a 30% failure rate. So if you're looking at urinary tract infection, that number climbs even to 67%. And it's not very surprising because you're taking a sample from an individual and you're transporting it to the lab. Some of those microbes already die in transit, and then you're trying to grow them in this little artificial setting of a petri dish. And we have multiple different types of bacteria and fungi there that have very vast different types of requirements in order to grow. And once something does grow, the misdiagnosis can be challenging because bacteria, they kind of look alike. There's some gram-positive, broad-shaped, catalase positive, negative, but you're using a phenotype in order to determine what these organisms are. So there's a high rate of misdiagnosis. For multibacterial infections like biofilms, plaque formations, or even wounds, you can only grow the planktonic bacteria. The majority of the biofilm is completely unculturable. While sensitivity testing is, of course, great in order to determine which treatment options are most effective, you can only test those microbes that you are actually able to grow in the lab. And then again, you're having them in a petri dish or in a growth media. It's a very different scenario than actual patient when you're administering the antibiotic. Based on DNA evidence, scientists have been sequencing all kinds of microbes for the last couple of years. We know that there's 10 to the 20 microbes living on this planet Earth. On every single one of us, there's 100 trillion microbes, and only 1 to 3% of those are actually culturable in the lab. So while a lot of them might be commensal, some of them are pathogenic as well. And in order to determine what they're doing for our health and disease, we rely on their genomic markers in order to determine who they are, what they're doing, and how to most efficiently treat them. PCR testing is a great alternative to culture testing. It's been from the 1980s. The benefit is that we are using molecular markers here. We're looking at the DNA or RNA of those microbes limited to a few targets. So you have to have somewhat of an educated guess what kind of pathogens you're looking for in order to select an appropriate target panel. And when it returns all negative, back to the drawing board, right? I would always recommend going for qPCR or quantitative PCR in order to determine how many cells are actually there. Is this maybe a contaminant? Are these just a few hundred cells, or are we talking millions of cells? So having the quantitative aspect of PCR testing can determine the clinical relevance of the pathogens identified. And then the other aspect, let's say you have two hits. You find two pathogens that are both present. Are they both at equal levels? Is one outcompeting the other? What is the biological meaning of finding those? So qPCR has a lot of advantages over PCR. And it is absolutely possible to determine molecular markers of resistance and sensitivity looking at the genome of these different bacteria. However, there are hundreds, if not thousands, of mechanisms of antimicrobial resistances, and with a PCR panel, again, you're limited to very few targets. And the situation is we have lots of pathogens that completely get undiagnosed. It can take weeks for fungi to grow in the lab, mycoplasma, ureaplasma, very challenging to grow. So we have all these severe pathogens causing infections in our animals, yet limited ability to diagnose them efficiently. The alternative is NGS testing, which very overly simplified, you can imagine like running millions of qPCRs in parallel. It's untargeted sequencing, but it is just as sensitive as specific as PCR. So no longer guesswork, is it a Pseudomonas, is it a Volcoria? You have DNA evidence in order to determine these different microbes. And because you beat the hell out of these samples, you can even diagnose biofilms as well. And the results are much faster, typically available in around 40 hours. But back to sea lions. As you know, abscesses are caused by a variety of different pathogens. We just heard that in the last talk as well. We see both bacterial as well as fungal organisms causing these abscesses, including Staph aureus, E. coli, Pseudomonas, Klebsiella. And we're learning more about the fungi that are causing abscesses, several species of Candida, Cryptococcus, and some PCR evidence has been coming up, pinpointing coccidioides as an important pathogen here as well. But these pathogens never exist isolated. We are all complex individuals, 100 trillion microbes on every one of us. And so having the idea of how these pathogens are influencing each other, maybe having co-infection, things like that, is what we can study by looking at the overall microbiota. And the microbiota is the consortium of all these different microbes in a given environment. And typically that includes bacteria, fungi, viruses, as well as archaea. And while parasites are not typically covered under umbrella term of microbiota, they significantly influence the presence of all these other microorganisms as well. There have been a couple of studies, not a whole lot, on the microbiome of sea lions, and so far none on the abscesses in these sea lions that we see in California. So what we did for this study, the aim was to characterize the microbiome of the abscesses in 30 sea lions. That was the sample volume that we had at the time that this abstract was written. And using NGS, we looked at both the bacterial and the fungal profile. And I did not have a video of a sea lion being sampled, but here you can see how simple the sample collection is. The clinicians were simply swabbing the impacted area and then broke this swab off into our collection media. So it's a non-invasive procedure. And the goal was to identify what type of pathogens do we see, both bacteria and fungi, and do we see some kind of ecological system network between these pathogens causing the infection? They may be elevating the infections amongst themselves. And people are usually a little like, what is NGS? I don't quite understand it. And so in order to determine a little bit more what this black box is, here's a quick video of what's happening in the lab. We break open all the cells, and then we have these robots that prepare the DNA to be ready to be sequenced. So it's very fast, hands-off, and very efficient. And then we massively amplify these DNA molecules. Of one copy, we have millions and millions of copies to then determine what kind of pathogen is present and align it back to databases. And when I say that we literally look at all bacteria and fungi, this is how we happen. This is here the spot where you can see the 16S and ITS region. It's like targeting a human thumbprint. We all have one, but it's specific enough in order to determine what this bacterial species is. And then we're going to have a quantitative aspect in order to determine is this a commensal, is it a pathogen, and ultimately returning a clinical report. So when we took these clinical reports that have been sent to us and looked at the overall microbiome composition, this is what we found for the bacteria. Every different color scheme represents a different phylum, and then within the phylum we have them color-coded to the genus on the left and the species on the right. And as you can see, the majority of the organisms we would find are firmicutes and bacteroides. We have a lot of diversity. Some of the organisms or taxa we were not able to identify down to the species level. And that's where it becomes a little more challenging, because a lot of research is more needed on these sea lions in order to determine what those species are. But even if we can't say this is the specific bacterial species, we can then climb the taxonomic ladder and at least determine the class, order, or phylum, and so on. The fungal profile is shown here, a lot less fungal diversity than bacterial, and that is something we commonly see in all kinds of different animal species as well as different sample types. And keep in mind, like an Malassezia cell is 1,000 times larger than an E. coli cell. So they take up a lot more biomass in the environment, and so it makes sense that we just see a lot less diversity. These are the averages abundances across all 30 dolphins, but it can be very skewed, saying one dolphin sea lion has 90% of an E. coli. So that would screw the overall relative abundances. And so we did an analysis of the core microbiome, looking specifically what are the organisms that are most dominant and present in the majority of the samples. So we had a cutoff of 20% of the patients, as well as at least 0.5% of relatives. And you can see some of the classics here, some of the organisms that I mentioned earlier, Pseudomonas, Staph aureus, Candida, and so on. And we can also see that there's a lot of other organisms. We have a mix of aerobic and anaerobic different types of bacteria. And what I personally find the most fascinating part is here shown in the co-occurrence analysis. We found 18 different bacteria and 3 different fungal organisms coexisted and building this ecological network amongst themselves. And I would like to draw your attention to the top right corner, where we have about 7 organisms that are all positively co-occurring with one another. That means on a statistical highly significant basis, if you find one of these organisms, you will find the other ones as well. And we see these as a polymicrobial infection. We have anaerobes right there, like Pseudomonas, which is an opportunistic pathogen, and we have anaerobes like Clostridium, Bacteroidalis, Streptococcacea, and Ruminococcacea. So this is already hinting at, just by identifying these organisms there, that this is tissue necrosis. And we most likely see a metabolic collaboration between those different organisms, in Pseudomonas degrading the oxygen, and then the other organisms coming in. We also see Citrobacter there, which is a fairly rare pathogen, but it is statistically significant even in our relatively small sample sets. So we can start seeing patterns of disease and the onset of these pathogens becoming more and more common. Then we have Clostridium in there, which is a toxin producer, and overall just highlights, you can't just see these infections and these abscesses as a single cause, caused by a single organism. We have a consortium of different microbes all collaborating together and further progressing the disease. Now, what is the clinical relevance of this? We have these complex microbial relationships, and it has a huge impact on the development and the progression of disease, and it has huge implications for the treatment decisions on whether to choose an antibiotic or an antifungal treatment as well. And NGS has the superior technology in order to identify multiple infection agents at once, at the same time, from the same sampler, ultimately with the goal to resolve these infections faster and more efficiently. And with that, I would like to thank my co-authors, Dr. Zapanta and Dr. Jacob, who are both scientists in my team, constantly looking for patterns of disease in all kinds of infectious agents. And while this study was on sea lions, we work with all types of animal species and all types of samples. And if you have any more questions, because I'm running out of time, I do have a table outside, and I would love to keep the conversation going. We are very research-driven, so we're always looking for data sets and even healthy reference samples that we can compare our clinical samples to. Thank you very much.
Yeah!